Today I'm going to talk about research from the Deshmukh lab at UNC on how mitochondrial DNA damage can cause axons, the long parts of neurons, to break down. I'll walk through the science in simple terms and explain why it matters. I designed this presentation for students who don't have a science background. I kept the language simple and explained any biology terms because this research connects to real issues like chemotherapy side effects that affect many people. To start, neurons are the cells that help our brain and nerves send signals. Each neuron has a cell body and a long axon that acts almost like a wire, carrying messages to other cells. In this study, the axon is the part that becomes damaged. Most people know that DNA is found in the nucleus, but our mitochondria, tiny structures that make energy, also have their own DNA. These two types of DNA behave differently when they get damaged, and that's important for this study. Why does this study matter? Chemotherapy drugs damage DNA to kill cancer cells, but unfortunately, they also damage the DNA in healthy cells. Many patients develop nerve pain, numbness, or tingling called chemotherapy-induced neuropathy. The researchers wanted to understand why this phenomenon happens. The big question was, if mitochondrial DNA, or mtDNA, in axons is damaged, does it cause axons to die in the same way that nuclear DNA damage does? And is a protein called p53, a protein important for nuclear DNA damage, also required for mitochondrial DNA damage? Next, we will see what the researchers used to test this hypothesis. The researchers used a special device that physically separates the neuron's cell body from its axon. This let them add a DNA-damaging drug only to the axon. That way, they could damage the mitochondrial DNA without touching the nuclear DNA in the cell body. In this study, the researchers checked whether mitochondrial DNA was actually damaged, whether the nuclear DNA stayed healthy, and whether the axon started breaking down. They also used neurons missing the p53 protein to test whether p53 protein was needed. Now we will look at the key results of this study. The first major result was that the drug successfully damaged mitochondrial DNA in the axon while the nuclear DNA in the cell body remained unaffected. This gave them a clean way to study mitochondrial DNA damage on its own. Next, they found that the axons slowly broke apart over a few days after their mitochondrial DNA was damaged. While the degeneration of the axons happened, the cell body stayed alive. This showed that the damage was limited to the axon, but was still strong enough to make the axon fall apart. If you look at the figures presented in this slide, they show that they were fully broken apart by around 120 hours. The most surprising finding was that removing p53 protein, an important protein for nuclear DNA-based cell death, did not stop the axons from degenerating. This means mitochondrial DNA, or mtDNA damage, triggers a completely different type of degeneration pathway that does not need the p53 protein. The results shown in the figures here show that the presence of the p53 protein did not protect the axons. The study showed that neurons have two separate responses to DNA damage. Nuclear DNA damage uses a fast system that depends on p53, while mitochondrial DNA damage uses a slower system that breaks down axons without involving the p53 protein. Why is the research that we just looked over significant? This research helps explain why chemotherapy often causes nerve problems. The mitochondrial DNA in axons may be damaged, causing the axons to slowly fall apart even when the cell body survives. Understanding this could help scientists find ways to protect nerves during treatment. Thank you for listening to my presentation about this research.